Good morning, dear students of class 12 science. We are dealing with the chapter Biotechnology, Principles and Processes. So this is the third part of this chapter. Okay, so we have already done the first theory part and the second theory part, and this is the third part of this chapter. So in the last class, we have learned about the enzymes. If you remember, we are dealing with the tools of recombinant DNA technology. And in the last class we have learned about the enzymes that are being used in this technology. So briefly, what we have learned under the enzymes, we have learned about the lysing enzymes, the clipping enzymes, which are exonuclease, endonuclease and the restriction endonuclease. Then we have learned about the palindrome and palindromic sequences. Then if you remember, we have learned about the nomenclature of restriction enzymes also. Okay, and similarly we have learned about the functioning of restriction endonuclease also. So all these things we have covered in the second theory or the second part of this chapter. So today we will deal with the second tools of the recombinant DNA technology, but before that we have got important questions. Okay, the question is that these restriction endonuclease are mainly isolated from the bacteria, or they are present in the bacterial cell. But how does the bacterium protect itself from the actions of this restriction endonuclease enzyme? Are you getting it? Because these restriction enzymes are present in many bacteria, but these restriction enzymes are not able to cut the bacterial own DNA. How? Right. So the answer is that in most of the bacteria, they have got a defense mechanism. Okay, they have got a defense mechanism to protect their own chromosomal DNA from the action of restriction enzymes. Because restriction enzymes will cut the nitrogenous bases of DNA molecules. Clear? So most of the bacteria, they have got a defense mechanism to protect its own chromosomal DNA from the action of restriction enzymes, and that defense mechanism is known as restriction modification system. Okay, so the name of that defense mechanism is restriction modification system. And how does these systems come into action? How does it will protect the bacterial chromosomal DNA from the action of restriction enzymes? So this defense mechanism has two functions. Number one, these enzymes restrict the propagation of any foreign DNA in a bacterium by recognizing and cutting it at a specific DNA sequence. So it will restrict the inhibit the multiplication of any foreign DNA in a bacterium. Number two is most important one. Okay, number two is the most important one for you, and the answer of these questions is easily given by this point number 2. So to protect the own DNA, bacterial chromosomal DNA, from the action of restriction enzyme, this restriction modification system will add methyl group. Very carefully, it will add methyl group to one or two bases. That means these enzymes will recognize the palindromic sequences. So these restriction modification systems will add the methyl group to one or two bases so that it will not be recognized by the restriction enzymes. Clear? So by adding the methyl group to one or two bases, the restriction enzymes will not be able to recognize its own chromosomal DNA. So DNA having the modified sequences cannot be recognized and cut by the restriction enzymes. So this is how a bacterium protects its own chromosomal DNA from cutting by the restriction enzymes. It's very simple. Clear? Simply, these restriction modification systems will simply add the methyl group to one or more bases of the palindromic sequences, because restriction enzymes will recognize the palindromic sequences and will cut it. So simply these systems will add the methyl group to one or more bases of the palindromic base sequences, and methylation of restriction sites and listen carefully. Methylation means addition methyl group, and this addition of methyl group or methylation of restriction sites, the bacterial DNA remains protected against the restriction enzymes. The restriction enzymes will not be able to recognize the bacterial chromosomal DNA, and hence it will be remain safe from the action of these enzymes. Okay, so the question is, how does a bacterium protect its own chromosomal DNA from cutting by restriction enzymes? Because these enzymes are present within the bacteria, but these enzymes are not able to cut down the bacterial DNA. How? So answer is there in point number 2. Clear? Okay. So now let's go to the second tool of recombinant DNA technology, and that is the vector. If you remember, try to recall back when we are learning about the recombinant DNA formation, we require a foreign DNA having the gene of interest, and we got a vector DNA. That vector DNA will carry the gene of interest, the segment of gene, right? It will carry that segment of gene and will lead to the formation of recombinant DNA. So now here we are going to learn about the cloning vector, which is one of the most important tool of recombinant DNA technology. So actually, what a vector? Okay, so the vectors are DNA molecules that carry a foreign DNA segment and replicate inside the host cell. Listen very, very carefully. The vector DNA, the vectors are DNA molecule. For example, plasmids we have learned in bacteria, which are the extra chromosomal circular DNA molecules. So the vectors are DNA molecules which can carry a foreign DNA segment having the gene of interest. Not only that, they are able to carry the foreign DNA segment having the gene of interest, but they are also able to replicate it. They are able to multiply it within the host cell. If you remember, recall that the recombinant DNA after its formation was inserted into the E. coli cell, and whenever the vector DNA used to replicate, their foreign DNA segment also used to replicate. Clear? So the vectors are mainly the DNA molecule that carry the foreign DNA segment having the gene of interest and replicate within the host cells by using the host polymerase enzyme. So whenever the vector DNA replicates, multiply simultaneously, the foreign DNA will also replicate or will multiply and produces multiple copies. Okay? So therefore, for transferring and cloning a foreign DNA segment, listen very carefully. For transferring and cloning a foreign DNA segment in a host or bacteria, carrier DNA is required, and such a DNA is called vector DNA or transport DNA or the carrier DNA. So we cannot simply put the DNA from one source into the body of the host. Clear? We cannot take the DNA segment from the donor or from the host and from the donor and we can simply put it into the body of the host. We cannot do it. We require in between a carrier, an intermediate carrier that can carry the foreign DNA segment and can put it into the body of the host. Clear? Simply, for example, like for going from Parking to Gantok, we require a vehicle, right? So here that vector is like a vehicle which will carry the segment of DNA from Pakyong to host, that is Gantok. Okay, so it's mainly intermediate, which is carrying the segment of DNA. So here, for transferring and cloning of DNA, we require a carrier DNA, and that carrier DNA is simply the vector. These vectors are mainly used to carry that segment of DNA having the gene of interest within the host, or else we are not able to insert that DNA having the gene of interest into the body of the host. Okay, so such a DNA is called carrier DNA or vector DNA or transport DNA, and afterwards we are going to learn the characteristic features of the vector also. Clear? Now the vector carries its own DNA along with the DNA that is foreign. Clear? So now we can see, if you can see over here, there is the gene of interest and we have got the extra chromosomal circular DNA molecule vector. Okay, so what we have done, if you try to recall, by the help of the molecular seizure, restriction enzymes will make a cut in the foreign DNA, and same enzyme will be utilized to make a cut in the vector DNA also, right? So when we make a cut over here by the help of the restriction enzymes, then therefore this vector DNA will look like this, the gap over here, right? So this segment has been removed from the vector DNA, and this is the segment which has been taken out from the foreign DNA. This DNA segment carries the gene of interest. Now what we do, by the help of the molecular glue, that is DNA ligase, we will insert this segment of DNA into the vector. Okay, so now this will be our this will be our gene of interest. Okay, so this is our gene of interest. Now over here you can see that a recombinant vector is being produced. Okay, why? Because it contains a DNA from the foreign source and has its own DNA. So this type of DNA is called recombinant, or we can say rDNA, or we can simply call it recombinant DNA, because it has DNA from two sources. One, its own DNA, and second DNA is from the foreign one. Okay, so this type of DNA is called recombinant DNA or the recombinant vector. Now whenever we will introduce this recombinant DNA into the host, let's say into the E. coli, which we have learned in our first class, if we introduce this recombinant DNA into the E. coli cell, okay, the host cell, now this recombinant DNA will multiply within the E. coli cell by the action of the enzyme DNA polymerase. So DNA polymerase of the E. coli cell will not only multiply the DNA of the vector, but along with the vector DNA, the foreign DNA will also be replicated and will produce several copies. So we can say that several copies of recombinant DNA will be produced when this recombinant DNA is introduced into the bacteria. Okay, so we have got different types of vectors like plasmid, phages, cosmids, and phasmids. But over here, the most important vector that we are going to learn is mainly the plasmids and the phages. Okay, so what are vector? They are simply the DNA molecules which are able to carry the foreign DNA having the gene of interest, and it will replicate independently within the host cells. Clear? And vector DNA with the foreign DNA will be known as the recombinant DNA or the recombinant vector. And whenever this recombinant vector are inserted into the E. coli cells or into the bacterial cells, they will replicate, they will multiply and produces multiple copies of recombinant DNA. So we have got different types of vector. Example we have got over here plasmids, right? We have got phages. We will not about bacteriophage, cosmids, and phasmids. Then we have got two artificial vector also. Okay, so first of all, let's begin with the first vector, that is plasmids. The plasmid vector is in the page number 585. Okay, but do kindly note this one also. Clear? So write it down, copy this one. All right, so what are plasmids? If you remember the bacterial cell, in a bacterial cell we will find the bacterial chromosomes. That is true. Bacteria don't have a well-defined nucleus. They don't have a nuclear membrane or the nuclear envelope, so the chromosomal materials will be freely floating inside the cytoplasms in the bacterial cells, and they will be packed in a structure known as nucleoid. This we have learned in class 11. Okay, try to recall it. And not only that, bacteria do possess an extra chromosomal circular DNA molecule also. Okay, they have got an extra chromosomal circular DNA molecule also, and that extra chromosomal circular DNA molecule in case of bacteria are known as plasmids, and that plasmids can be utilized as a vector in case of biotechnology. Okay, so what are plasmids? So plasmids are small extra chromosomal self-replicating circular double-stranded DNA molecule occurring in bacterial cells along with the bacterial chromosomes. So bacterial chromosome is there in bacterial cells, but along with the bacterial chromosomes we'll find the plasmids. So if I will draw it roughly, if I am drawing the cells of bacteria over here, here you can see the presence of bacterial chromosomes, okay, which are freely in the cytoplasm because there is no nuclear membrane. So we can say that bacteria do not possess a well-defined nucleus, meaning there is absence of nuclear membrane. But along with these bacterial chromosomes, we will find a small extra chromosomal circular DNA molecule in bacterial cells. So the small circular chromosomal DNA molecule, this one, okay, it's known as plasmids. Now this plasmid has one more advantage. It replicates autonomously, independently. Clear? It replicates independently. So it replicates autonomously, independent of replication of the bacterial chromosomes or division of bacterial cells. So it has got an independent way of replicating. It is not dependent on the bacterial chromosomes for its replication to produce multiple copies. So it can replicate independently. It can produce multiple copy. Clear? So although plasmids are usually not essential for the normal cell growth and divisions for the bacteria, but they used to provide an important trait. TRAIT traits means character. Listen carefully. TRAIT traits means character. Okay, so these plasmids, though they are not essential part of the cell, they are not recognized for the cell growth, alright, normally, but they used to give the bacterial cell an important character, an important trait, alright, to the host organisms. And what can that be? Examples: resistance to certain antibiotics. These plasmids can confer the bacterial cells the resistance to antibiotic property. Okay, that means if this bacterial cell has got resistance to antibiotic, let's say penicillin, just for example I am saying, if these plasmids will confer the resistance to antibiotic penicillin to the bacterial cell, then that bacterial cell will resist the action of penicillin. It will not die when we are using the penicillin. That is called antibiotic resistance. Okay, so these plasmids will confer antibiotic resistance property to the host cell. Clear? So one of the most easily manipulated plasmid vector that we are using is plasmid PBR322. This is very, very important. Can you remember? The most manipulated and easily utilized plasmid vector is PBR322. Okay, so in the name of this one, the letter P stands for plasmid, B and R stands for the initial of the two scientists who discovered it. B means Bolivar and R means Rodriguez. Okay, who developed this vector PBR322. So now let's see the few features of this vector, or the vector PBR322. So can note it down. So the plasmid contains ORI site. So look at the figure also. Can you see over here the ORI site? ORI site means origin of replication sites. So this ORI site will help in the replication or the multiplication of recombinant DNA or the foreign DNA. Clear? Next one, it has got two selectable markers. So over here, the selectable markers are AMPR. This AMPR means ampicillin resistance and TETR means tetracycline resistance. Okay, so this AMPR and TETR act as the selectable markers. We will learn afterwards what is the function of selectable markers. Clear? Then it also has unique recognition sites. Clear? It also has unique recognition sites for the 12 restriction endonuclease enzyme. Clear? So it has a unique recognition sites. So what are those unique sites? If you look in the figure, you can see PST1 and PBU1. So this PST1 and PBU1 are the unique recognition sites which are present on the region called AMPR. And similarly on the TETR also we have got unique sites which are BAMH1 and SAL1. These are the unique recognition sites which are present on the vector PBR322. Clear? Okay, so don't confuse that. AMPR means ampicillin resistant site and TETR means tetracycline resistant site. Alright, so kindly go through this diagram. It is very, very important. And this diagram you can see in the page number 586. Alright, page number 586. You can see the figure over there. You can see the vector A PBR322. So kindly go with the figure. Alright. So this is the first vector. Now we will learn about the second vector. The second vector is bacteriophage vector. So kindly note on the first line. Okay, the first point. Now, what are bacteriophage? because we are going to learn about the bacteriophage. So you should know what are bacteriophage vectors. Clear? So bacteriophage are the viruses that can infect the bacterial cells. It's very important. Bacteriophages are the viruses that infect the bacteria by introducing DNA into the bacterial cells. So these are the viruses which can infect the bacteria. And two phages that have been extensively used and has been modified for the use as a vector are phage lambda and M13. These are the two types of bacteriophage which has been modified to use it as a vector for transferring the DNA into the host cells. Okay. So bacteriophage or the phages. Lambda phages are also used for transferring genetic material from one bacterium to another. So their DNA is linear double-stranded molecule of about 48.5 kilobase pair. It contains several target sites for the restriction enzymes, and phages are better vectors. Why? They're small in size and they can transfer large DNA molecule or segment. Number two, they can be easily detected at the time of cloning experiments. Similarly, the third vector that we can use is the cosmids, COSMIDs, cosmids. Okay, and cosmids has also few advantages over plasmid vectors. Number one, it can transfer a large foreign gene ranging from 40 to 45 kilobase long into the bacterial cells. It can be used in the preparation of gene bank of small as well as large sized organisms or genomes. Cosmids can also help in the study of some nonsense sequences. Okay, so this is the importance of cosmids. Then we can also use the phasmids, PHASMIDs, phasmids as the cloning vector. Okay, so these are the plasmids. So phasmids are the type of plasmids with a fragment of lambda phage DNA along with the attachment sites. Okay, so generally among the four types of vectors, plasmids, bacteriophage, phage or let's say lambda vector, then cosmids and phasmids, the two are most important one. Okay, first one, you will go through the plasmids, and in plasmids you will go through the structure of the vector PBR322. We have got the ORI sites, we have got the ampicillin and tetracycline resistance sites which act as selectable markers. Then they have got few unique recognition sites. So that diagram is very important. Okay, this diagram is very important. You will go through this diagram. Then second important vector is bacteriophage vector. Okay, and what are bacteriophage? They are the viruses which can usually infect the bacterial cells, and now they have been modified to be used as a vector to transfer the foreign DNA into the bacterial cells, and few modified bacteriophage was mainly phage lambda and M13. Then you have got cosmids and phasmids, but most importantly you will go through the plasmid vector and the bacteriophage vector. Clear? So next one, the next topic, that is characteristics of cloning vector. We will deal with that in next class. Okay, so today we will simply go with we have gone through the vectors, we have gone through the four vector. Okay, so we will go just kindly go through it. Everything will be clear for you. Okay, so in the next class we will begin with the characteristic features of the cloning vector. Clear? So see you in next class.